So basically, I'm going to talk about something that is really should be off the beaten track of all of you, which is the use of deoxy-hemoglobin as a contrast agent. It was off the beaten track for us too. This has been a crazy, weird trajectory for us. And, you know, I've consulted Alex and John over there and a number of others who know about this. And it's certainly not a fully baked cake yet. It's half baked. I'd be very happy if people who are, I'm going to present to you as far as we got. And if you have any comments or giving us some hints, I would very much appreciate that. Okay, how do I go forward here? Or just use the keyboard?
Yeah.
All right, so disclosures. I'm the founder of this company called Thornhill Medical. We make a number of goods that are basically military. This is carbon monoxide treatment. This is a life support system that is being used by the US military and other militaries around the world. This is an anesthetic machine that, you know, usually an anesthetic machine, if you've been in the OR, is about the size of a small refrigerator. This one is the size of a 2 bread toaster. It's about this big, and it's completely portable and can be used in the field. So where does this come from? Well, this is, you know, we had all these good engineers in the company, and I was interested and was working with Dave Michaelis looking at brain and MRI, and we needed something to control the PO2 and PCO2. So I used my influence to get the engineers to make this. It's not a commercial device, but it's available for basically to support research and basically just recovery of costs is basically what it is. All right. So what about the gadolinium? When a bolus is given of the gadolinium way to the heart, you'll recognize this from the anatomical likeness, and it goes along the vein towards the heart, and it's mixed in with other veins that come from other parts of the body, progressively diluting your contrast agent. It goes into the heart, gets washed out from the heart, and gets progressively dispersed until it finally gets to the brain. And this is basically the gadolinium going through. You're all familiar with this, I'm sure. And this was sped up for time, but it takes about 10 seconds for this to come through. In contrast, it took only 1 second for the injection. So the rest of it is this dispersion, which you get along the way. So, so once you have the gadolinium, your contrast agent there, you need to analyze it. So this is the well-mixed model where your contrast comes into the voxel. You have a mixer, and the model, the fluid inside with the contrast is displaced out. There's other papers which give you this type of model, which a bunch of blood vessels come in from a blood vessel and end up going out. So the model for analyzing that, and for us, you will see that there's a step change in the contrast agent. But the model was that the change in the signal is your cerebral blood volume. And if you did the first derivative of the upslope here, you get a peak flow, and that's cerebral blood flow. And the time to peak is the mean transit time. This is what we had to work with because this is what was in the literature, but we didn't totally buy that. In any event, talking now, coming back to about deoxy-hemoglobin, which is the what we wanted to investigate. So these are the lungs, and what we thought was we would replicate the gadolinium with deoxy-hemoglobin. So you have oxygenated blood here, you have deoxygenated blood, and then we would come back to the oxygenated blood. The interesting thing, we did this back in 2000, in about 2000, is that these little red dots are the breaths, the progressive breaths. So it takes a number of breaths before you can reduce the saturation of the blood and get your deoxy-hemoglobin. And then when we reoxygenated, there were literally 2 breaths. So that caught our interest. We looked again, and this time we decreased the TR to about 200 milliseconds. So hypoxia, boom. Hypoxia, boom. And we expanded the time here, and it takes about, this is about 101 seconds, and by about 102 to 103, boom. At 200, by 201 or so seconds, boom. A second. Crazy. So why does that happen? Well, this is how I was taught the lungs look like. These are the trachea, and then these break the smaller and smaller bronchi until you get way out into the periphery where the gas exchange actually takes place. And the lung looks like that to an anatomist, but to us, it'll look like this. This is a model where the cross-sectional area is here, and the length of each part is over here. It gets shorter and shorter, but the cross-sectional area starts to get bigger and bigger. By the time you get down to the 19th or so branch, it starts, you get the acinar alveoli, and they start to be able to diffuse some oxygen into the blood. But by the time you get to the 23rd, so just another 4 branches, the area goes to 130 square meters from 1 square meter. And the time for the gas to get from here to here is milliseconds. Milliseconds. So if you put oxygen down here, this blood coming through stays deoxygenated, the air, the gas that you inhale gets over here, the oxygen gets here, the oxygen gets here, and then bam. I don't want to use just technical terms, people aren't familiar with that. Yes, bam is right. And this is from Alex Bogal. This is hypoxia. And then here it's reoxygenated, bam, watch it again. Hypoxia, bam, it's reoxygenated. This is the part that we found very interesting and decided to investigate a little bit further. So I just want to point out about the analysis of this. So with the well-mixed model, this is continuous exponential. So you use arterial input function, you look back, you deconvolve it to see what went in, and it comes out. If you look at this model, this is not the same model as this, even though the analysis in the literature is about the same. What happens here is that once the blood gets into the blood vessel, it doesn't matter how many vessels and how long they are and how long it takes them to leave. As long as the blood is coming in before any leaves, there's a straight line. And I have a slide at the end of this talk where I can go into this a little bit further, because this was very puzzling. It took us a long time before we were able to swallow this, because it's so contrary to what is told. But the going out of the voxel is an exponential, because some will traverse the voxel very quickly and some will take some time. So there's the voxel, and here's the model. So initially, okay, I drew these, so I apologize. I tried to trace it from stuff that was in the literature. So the red are is now the oxygen, bam, from that slide. So the red is the oxygen, and they don't arrive all at the same time. You can see some vessels have still not arrived, and some are already percolating through the voxel. And that's what happens between A and B. By the time you get to B, all the blood vessels entering the voxels are inside, and you get a linear response, as I just showed you in the last slide. Okay, if there's any question about that, you can ask me during the question period. And then here, I took off the roof off the voxel. I'm no artist, obviously. I should stick to what I'm doing. And you can see that some of the oxygen is now leaving the voxel. So there's a decrease rate of increase of the signal. The rate of increase in the signal is now getting lower and lower. So how to analyze this? Well, there's some reference time, and this is the relative blood arrival time by the time you get a noticeable change. That's okay. There's the linear portion. You can draw a line through that linear portion. And the slope of that line is the cerebral blood flow. How do I know that? Because all the vessels are now in the voxel, and the rate of flow into that voxel from all those vessels is the blood flow, the relative blood flow. So now if you take a tangent to the curve here, this is an exponential curve, take a tangent to the curve, and this is one time constant. And you do the same thing, you draw a tangent to the curve, and this is another time constant, and you add the time of transition during the linear portion, and that's your mean transit time. How do I know this is true? Because this is verified with the central volume theory. Because here is your the cerebral blood volume here. Here's your cerebral blood volume over the mean transit time and your slope. There's your central volume theorem. And if you look at it writ large, it's there in the same way. Goes down from here, down to here, and across to where this line comes, and there's your central volume theorem again. So the central volume theorem verifies this analysis. Now, that's a little thing, that's a little step, but my God, was it ever hard. You know, John will remember we probably spent, a whole hour just there, arguing it through, and with Alex and with everybody else. It was hard. So what did we get from it? So this is a healthy patient, and this is the gadolinium. These are the mean transit time, cerebral blood flow, cerebral blood volume. And I just appeal to your looking at these and seeing that they're both pretty well the same. This is a little bit easier to understand. This is a patient with middle cerebral artery stenosis. And you can look at the mean transit times, right? And the relative cerebral blood volumes are also have the same pattern. But I'll show you something else, which I think is also very convincing. So this is a series of slices, because here we show you the same slice. Now we're going to show you a series of slices. This is deoxy-hemoglobin, but not the step change. This is our arterial input function. Look at the evolution of the contrast here, and compare that to the hypoxia, and you'll see that overall you'll get the identical changes. You may be one slice away or not, but you get the absolute same changes with each of them. So the transient hypoxia versus gadolinium, it's got a few advantages. There's no injection. You can repeat it as many times as you want. You don't have the gadolinium. You don't need a blood-brain barrier. All the deoxy-hemoglobin is intracellular and doesn't leave, so it's completely repeatable. And it's got some disadvantages because we decrease the saturation to about 75%. People are quite hypoxic. But having done it to myself and to all of us in the lab, it's very tolerable. Most of us can't sense any change at all. The gadolinium has disadvantages. You have to inject it. It has the advantage perhaps of signal to noise, or it could be that the signal to noise is what we find saturates, and you don't get the full range of changes that you do with deoxy-hemoglobin that is much less. And it's got some disadvantages. I'll show you this disadvantage here. About 200 tons of gadolinium are peed out by patients who get it injected into them and eventually makes it to the ocean. And this affects fish, shelled animals, and sea urchins, and it mutates them. Gadolinium and mutation. There's no pollution at all. This is what it looks like in our MR unit. It's fairly straightforward to set up and help save the oceans. Some things that you think are impossible are just inconceivable. They're not necessarily impossible, which is how I made my career. And I'd be happy to, I would love it if you can put up your hand and say something and point out something to me that you think that we're not doing right or that we could do better. I brought my notebook down here to take notes, and I'd be happy to answer any questions otherwise.